I'm going to record, and I will do a short introduction of you, Peter, and then I will pass you to start sharing the screen. Okay?
Okay. Let me test sharing the screen. Okay.
Okay, everyone. And today we have Péter Poczai. He's a doctor of plant genetics, working as curator of the botanical collections at the Finnish Museum of Natural History of the University of Helsinki. Peter has been research fellow at Radboud University in Netherlands and a scientific authority in plants from the CITES. Peter research lies in the area of plant molecular systematics, molecular evolution, and comparative genomics. His research includes a range of experimental approaches, from the collection and observation of species in the field to the generation of large genomic datasets. He is especially interested in wide relatives of chloroplants and their use in plant breeding. Most of his work involves the large and economically important nightshade family, Solanaceae of course, including potato, tomato, and eggplant. And today he's gonna give us the talk entitled Plastid Genome Evolution in Solanaceae, Past, Present, and Future. Thank you, Peter, for being here and to accept this invitation. Please, if you can share your screen and start your presentation, you're welcome.
Yes. Hello, everyone. I'm going to share my screen now. Okay. So here we go.
That's perfect.
Yeah. Is it totally visible? Yeah. Okay. Then I could start now. So welcome everyone. I'm a bit excited. I'm not going to be. I'm quite shy in this term, but I'm trying to give you an overview of plastic genomes in general, and one of my favorite topics and favorite plant families, of course, Solanaceae, and this is why I'm here. So our menu for today is just to be on the same page with plastic genomes. I will give a general overview and give you some highlights of plastic genome research and what has been going on and what are the key features and key components, and then we will start talking about Solanaceae plastic genomes in general, and we will go through its evolution, their evolution and structural features, why they are important, and why should we observe them, and why they might have some biological relevance in terms of phylogeny and phylogenomics, and we will look into some specific features of repeats and positive selections. And I'm trying to continuously highlight different things plastic genomes, and let's see what it has been and what has been done in the past. It is quite this topic is very precious to me because most people, I grew up on the Richard Palmer and Olmsted papers where you saw all the all beautiful plastic genome stuff, and then somehow it stuck with me, and then I'm into it. Okay, hope you will enjoy it. So the first parts, why are plastids so important in general? So plastids and chloroplasts and plastids as a whole are eukaryotic organelles, and their pigments alone provide the world with much of its beautiful and mellifluous colors, not to mention their clever and life-sustaining ability that they can convert carbon dioxide and water into oxygen through a quantum mechanical process, which we all know that's photosynthesis, what I have highlighted in the upper corner. Plastids are also the engines primarily responsible for generating the fruits, vegetables, and grain we eat, basically the basis of plant life. Plastic-containing organisms are also seeding the world skies with clouds, providing us rain. As we see down here, the single-celled algae called Emiliana huxleyi in a recent paper was highlighted as which is actually the ocean's most abundant microalgae, a microorganism, provides the kernel on which water vapor can condense, and then clouds could be formed, and then that is providing us rain besides Pseudomonas bacteria. These compact cellular organelles, of course I'm talking about plastids, are also preserving very well in dry plant herbaria and dried plant material for centuries. And this is why it is allowing us to investigate important evolutionary biological questions in terms of museomics research. Besides important conservation and taxonomical questions, what I will emphasize later, but the most intriguing thing about plastids is where do they come from? So the landmark event of endosymbiosis leading to the big group of Archaeplastida, or what we call plants, are arising from the primary endosymbiotic event, which conferred on eukaryotes the benefits of photosynthesis, and in so doing had a huge impact on the course of evolution and the climate of Earth. It also led to an extinction event. Before this event, photosynthesis was solely a prokaryotic trait, and in a way it still is, since eukaryotes and plants never learned how to photosynthesize on their own. Regarding their origin, there are majorly two hypotheses. The first hypothesis, which is the freshwater hypothesis, argues that the first endosymbiotic event occurred in a low salinity environment. Both red and green algae colonized marine environments early in their histories, with personified green phytoplankton diversifying around 85, 65 million years ago. There was also a very nice study which published the discovery of the Gliomargarita lithophora, which shuffled a bit the evolutionary tree of cyanobacteria and also the origin of plastids. While the other hypothesis, the nitrogen fixing hypothesis about plastids, states that the nitrogen fixation, along with the chemistry of photosynthesis, was an early driver for endosymbiosis, but became less important to the partnership with increasing nitrate abundance. In this context, the rise in atmospheric oxygen could have been the driving force that promoted nitrogen-fixing cyanobacteria to invade microaerobic environments found in the cytosol of a mitochondrial phagot trough, so as to protect their nitrogenases against oxygen inhibition. The hosting cell may have been benefited from the carbon and the nitrogen-fixing exudates from this endosymbiotic event. Of course, there's a lot of talk about these hypotheses, but when this event happened, you will see the summary of the current research and current phylogenetic trees about placing plastids on the evolutionary tree. It's not an easy task. As already mentioned, cyanobacteria are the origins of plastids, as most of us know, through a unique event of the endosymbiosis. Consequently, plastids form and should form a morpholytic group within cyanobacteria. Although this is well known in the scientific community, the precise position of plastids and when this extended diversity of cyanobacteria has been a matter of discussion in recent literature. Two major scenarios are opposed, one postulating an ancient origin of plastids. This is called the early branching hypothesis, while the other one postulates a relatively recent origin. The hypothesis of an early origin around clade E among these trees, you will see it with with this interesting bluish color is nevertheless more frequent in literature and has been more recently strengthened by the study what I mentioned on the early on Gliomargarita lithophora. So yes, as the closest which turned out to be the closest extant relative of plastids. In this context, the early branching origin, their exact position is not complexly resolved. Indeed, plastids appear to be interior and to different groups, most likely to the mentioned clade E, which is the eukaryotic group. In very early branching scenarios, in a majority of studies, but they are posterior to clade E. So plastid positions are swapping quite frequently on current phylogenetic trees. What is the solution to this phylogenetic question? That's remained to be answered. What is most important from a Solanaceae perspective, of course, is that thanks to the symbiosis, we could study and utilize organelle genomes because they have their separate own DNA compartments. In this respect, Solanaceae was a very important plant family since the first Plastid genome sequence was published for Nicotiana for tobacco back in 1986. But we shouldn't forget about the liver for chloroplast DNA sequence, which was also published in 1986 in a different journal, though. What is quite remarkable about these two figures is that the tobacco genome depicts a linear plastid genome, while the liverwort chloroplast DNA is represented by a circle. And the two theories and the two things are still going on in recent literature. And if you are publishing your paper, you most probably see the circular forms more frequently, and that is dating back to 1986 with the Marcantia polymorpha genome being sequenced. But to be on the same side with plastids, as we see, they are very compact cellular organelles with their own DNA, and they possess unique quadripartite structure, which means that they have a large single copy region which is separated by two inverted repeats, which should have identical sequences, and the two sequences are getting homogenized through gene conversions. And there is, of course, our little small single copy version, the SSC, which is separated by the two IRs. Basically the IRs depict in a circular form or in a linear form as well the junction sites, which will be very, very important in terms of evolution as well. Plastid genomes include 110 or 130 genes, primarily participating in the photosynthesis, transcription, and translation. And their conserved gene content, order, and organization makes them relatively well suited for evolutionary studies, since gene losses, structural rearrangements, and pseudogenes, which are characteristic for Solanaceae, are additional mutation events which could be characteristic for certain groups during evolution. As we see that subsequent studies incorporating an array of tools have found that these genomes comprise complex assemblages that include circular molecules, but where large linear and branched forms predominate. So if we look at the plastid genome under an electron microscope, we would see this structure with a circular structure in the middle, with different linear branching forms coming out of it. And these are recombining and shuffling with different intermolecular mechanisms. These mechanisms, which facilitate the replication and the repair of the organelle genomes, are inherent to their dynamic structure. So rather than the plastid genome being a conserved cellular organelle, it could be rather depicted as a rather dynamic than a dynamic evolutionary process. And a recombination-dependent replication, which is a mechanism behind the homogenization of the plastids, and this mechanism is also diversifying plastid diversity. So, what is the next slide? I would like to show you something which is not necessarily specific to Solanaceae, but maybe it is less known in Solanaceae, which is the inheritance of plastids. So the transmission of the chloroplast genome is mostly attributed to the maternal inheritance. The transmission of paternal chloroplasts was observed in Nicotiana tabacum under stressed conditions. It was in the 80s. It was done in vitro. It was accidentally discovered in vitro cultures at the University of Szeged in Hungary, when plants carrying streptomycin resistant plastids were used as pollen donors, and cell lines with paternal plastids in the offspring were selected as green resistant sectors of induced callus, and they included from the seedlings and the streptomycin containing media. Of course, low frequency paternal leakage of plastids via pollen was suggested to be a universal phenomenon in plants, with strict maternal plastid inheritance to be expected. But the paternal transmission of plastids was also observed in other Nicotiana species, more specifically Nicotiana plumbaginifolia, with a bit higher frequency, which reached almost 15% in some studies. But in all cases, plants carried maternal mitochondria. So further studies investigating the paternal origin of Nicotiana species should also focus on mitochondrial genomes, and which would exclude possible low-frequency paternal plastid inheritance, and investigate the paternal inheritance of plastids in Solanaceae in general, because paternal inheritance can be triggered by stresses, which could be very important in many ways in Solanaceae evolution. So after this general introduction, that's how we know it from the 80s, we move on to the high throughput sequencing age and the DNA library preparation, which dramatically improved how plastid DNA can be obtained at low cost. Nowadays we reached that one plastid genome sequence could be sequenced around 100 to 200 euros each, with a very nice coverage. And we can thank this to different high throughput sequencing machines, what I guess most of us use in our research nowadays. So this led to a dramatic increase in plastid genome sequences deposited in NCBI, also which is this is back to 2017. And since that, the number of sequences is just simply increasing in NCBI. This is very, very important, because this led to some people to postulate that plastid genomes could be used as kind of super barcodes, which is an extended version of plastid barcoding, since most plastid regions and plastid genomic regions, mostly IGS regions, intergenic spacer regions, were used as barcoding regions. So could we use the extended entire plastid region as a universal barcode for plant evolution? Let's see some specific Solanaceae genome features in this respect. So if we look at the Solanaceae evolutionary tree, what is striking is that only 160 plastid genomes have been published so far. We have published quite substantial amount of this one. But what is quite striking, if we look at the tree, is that 55% of all the published sequences are coming from the genus Solanum. So what we see here is the typical crop species, and species getting sequenced which have economic importance. So, but other species have less sequences available in NCBI. If you look at the larger picture, we see that 99% of the available sequences are coming from the so-called X12 clade, and only 2 sequences are available from the early branching groups from this basal part of the evolutionary tree. We have only 2 Petunia sequences. So if you are out there, I can just simply, and you want to explore or want to make a good discovery, just sequence all these things found in the basal group. I can assure you that you might find something very, very interesting happening in the basal groups and the basal node and the first branching group of Solanaceae evolution. So this is what we have at the moment, and not surprisingly, we could use these sequences to study different evolutionary genetic processes or evolutionary processes of certain groups. There were some previous talks before my talk, and specifically Edlin's talk, where you saw that if you would try to use plastid genomes as a whole, you might get large polytomies in your trees, and you may not get a fully resolved tree. But if you are concentrating on smaller evolutionary level or smaller evolutionary scales of specific groups, you might be able to uncover interesting scenarios. For example, Xavier's work on eggplants, I guess you might be all quite familiar with that. I had the pleasure to contribute to that. It was pure joy working on this thing. Which was the major finding of this study was that using plastid genome sequences, we were able to infer the evolutionary scenario of eggplants, which were interestingly coinciding with the distribution patterns of larger mammals, which revealed impalas and elephants to be the major distributors of the wild relatives of eggplants. So if you would like to conserve crop wild relatives of eggplants, then you should also consider conserving elephants as well, of course across Africa. The other important thing what I would like to highlight on the other side of this publication is that plastid genomes are usually generated by larger sequencing initiatives, but they are discarded as contamination, and so in so doing, many interesting biological phenomena are ignored. So, for example, if we look at tomatoes, and we infer the phylogenetic tree based on nuclear data sets based on whole annotated transcriptomes, we will find that Solanium hypocrites and Pennellii are appearing together as related at a very basal position at the phylogenetic tree. And when we infer the plastid tree, we would see two different plastid clades. We would see an age group which is characterized by Solanium hebrochites type of plastid and related species, and then we would see different species where Solanium Pennellii appears to be basal, and these plants have plastid genome structure, which is much more similar to the repeats and the microstructural changes found in Solanium Pennellii. And these biological phenomena were more or less unknown because they were just simply discarded by sequencing initiatives. But they could have very important agronomical and practical implications, for example, in grafting. So as we see, and I told you, Solanaceae, and if we look at the junction sites of plastid genomes, which are depicted by the inverted repeats across different Solanaceae, what is quite striking is that Solanaceae plastid genomes are highly synthetic. This doesn't mean that they would be boring. They are just simply, from a molecular evolutionary point of view, they are not as exciting as a highly rearranged plastid genome of Pelargonium or Silene or some other RAC or aroids. But Solanaceae plastid genomes are also very excited. But if we would like to discover these properties, we would have to look for in other parts of the genome. What is quite interesting is lies in the genome annotation of Solanaceae plastid genomes. We see a flip-flopping GLB, which is a connecting junction site of the large and of the LSC and the IRB, and we see a very small truncated pseudogene of the RPS9, and which is also getting copied at the other part of the connecting junction, which lies between the tRNH and the PSBA gene. The tRNH and the PSBA IGS region is well known as a common plastid genome marker for phylogenetic studies. And in Solanaceae, the tRNH gene is always located in the LSC and then the other side of the GLA. So far, no rearranged plastid genomes have been found. But as I told you, you should look into the first branching groups of Solanaceae, and you might find some interesting features. So if Solanaceae plastid genomes have a highly synthetic structure, then does that mean that they are uninteresting, or should we just simply abandon the topic? No, simply no, because that means that besides nucleotide diversity and due to this highly conserved structure, more or less the the intergenic spacer regions are also synthetic. So they could be utilized as indel markers in different barcoding initiatives. And based on Plastid genome information, if we look at the sequence of Solanum scabrum, where we are comparing different close related groups, so Solanum scabrum and their relatives are highly similar plants. They are very closely related. They form a polyploid series, and they are very hard to deal with, and they are very hard to differentiate. But if we try to look at the nucleotide diversity, what I have presented with our tool, recently developed tool called Chloroplast, I could just recommend you trying to use this tool, because with this bioinformatic interface, you are able to depict the hotspots along the chloroplast genome. So the outer ring, we see the indel diversity of the different regions, while in the inner ring, we see the nucleotide diversity. So we see that while nucleotide diversity is very low in many cases, but indel diversity is much higher. And based on this phenomena, we are able to differentiate and barcode the species. The other example what I would like to see, which also relates to barcoding, is coming from Britannia. If you are working with Britannia systematics, please somebody look at the herbarium specimens. We need that. We really need that, because many visiting PhD students of mine recently performed a study, and we found many interesting barcoding regions, because Britannia and mostly Britannia somnifera products and root extracts are coming mostly from India and Pakistan region, coming to the EU and also to the US. And it is very hard to know what they are formulating inside these vegetable capsules. So, but to be able to separate and comply to EU regulations, or maybe even to US regulations, we have to find an easy method to differentiate between these species. And Plastid genomics could be a very easy and handy way to differentiate these species. So if you are thinking about Plastid genomes in terms of Solanaceae, and you have the same issues, I could just advise you to go on and try to perform these kind of studies. I think Plastid genomes will be a good choice for you. But as I mentioned, Solanaceae Plastid genomes are highly synthetic. So this means that every little detail and change could be very characteristic in an evolutionary sense, and it could be used as an additional support for phylogenetic purposes. As we all know, for example, these kind of solid evidences, like for example, which is uniting the X12 clade, could be also found in different phases. So one remarkable thing is, of course, the presence of phenylalanine like TRNF pseudogenes, and also based on these interferences, you are able to reconstruct the evolutionary processes of different genes, which could be very important in terms of adaptation or in terms of adaptive radiation, or occupying a new evolutionary niche. So let's look at a bit closer the repeats and different selection pressures acting on Solanaceae plastid genomes. This could be a hard figure for the first time, but I try to emphasize and try to describe what I would like to tell in here. So repeats in the plastid genomes, other than the non-IR containing repeats, are very important in terms of recombination dependent replication and may initiate through strand inversion at non-allelic sites. Depending on the orientation of the repeats and the specific repair mechanism employed, novel repeats, inversions, and deletions. On the other hand, rearrangements can occur in the plastid chromosome, where non-allelic repeats employed by repetitive repair mechanisms lack complete nucleotides. So gene conversion, when I mentioned for the IRs, can homogenize the repeated sequences, favoring either a native or a novel base at any site along the length of the repeat. But replication initiation at a non-allelic repeat could lead to completely new chromosomal arrangements. While most plastomes lack larger than 100 base pair repeats, recombination reactions have been demonstrated to initiate at microhomologous regions where expression of nucleotide encoded DNA maintenance proteins have been impaired. This is very, very similar to what we see in plant mitogenome containing repeats. That is also characteristic, for example, large fungal mitochondrial genomes that provide substrate for further recombination, and this recombination generate diversity and generate continual structural variation within the cells, within the same tissue, within the same individual, and this process alone could also be the leading source of heteroplasmy with respect of the organelle genome structure and nucleotide substitutions that arise in the context of nucleotide that uninvolved aggregation of proteins and nucleic acids that each contain multiple organelle genomes, genome equivalents, and this variation may be heritable. So a good example of this repeat-mediated recombination is the generation of pseudogenic region in a specific Solanaceae group, so which are called pseudosolenoids, which happened in the early ancestor of this clade. And so far, all members which have been sequenced from this larger group contain the remnants of this repeat-mediated structure or parts of tRNA pseudogenes. Which was created through repeat recombination, and this is also specific to different species and to also different populations, and it could be also variable because different species have different contractions and different parts of the same pseudogenic particle, which is majorly composed of a T loop or a variable loop, and then of course a D loop and an acceptor stem. Most of these leaf clover parts of the tRNA and the pseudogene tRNA particles are lost during the evolution, but what is quite common is the conservation of the anticodon loop, which is preserved in this in most Solanaceae species where pseudogenic particles, pseudogenic parts are found. So this is quite important. why these molecular all the processes when I'm talking here are are all molecular evolution and molecular processes that could be linked importantly to a phenotype. So as if you haven't read Stacy Smith's paper in Trends in Ecology and Evolution, is that phylogenetics is the new genetics, at least for biodiversity. You should definitely lead that, because the most important thing is linking these molecular processes to a phenotype and to adaptation, and put them into a biological and evolutionary context, underlying and pinpointing different evolutionary processes, which would be very, very important for all Solanaceae research, right? So let's see another example. I would rather go back here. Okay, so we talked about repeats, and we know that Solanaceae plastid genomes are highly synthetic, but a lot of important things are going on in the IGS regions and in the non-coding parts. But besides that, besides a possible paternal inheritance, the other important thing could be with Solanaceae plastid genomes is positive selection, because recently the plastid genome was regarded, and plastid protein coding plastid genes were regarded as a highly conservative structure with different parts that are under purifying selection or negative selection, if you want to put it like that. But recent studies have shown that this may not be the case for many scenarios. A couple of years ago, only the number of we published one paper in 2018, and back then, when we were working on it, there were only maybe 5 or 6 papers that reported positively selected protein coding plastid genes. And since that, there was a very nice, interesting paper published in Planta by a French group, which showed that actually in Poales and grosses, in Poaceae, most of the protein coding genes could be under positive selection, and they could have important functions. And this could also lead to a phylogenetic context. So if you have plastid genomes and then a plastid genes and an entire genome, treating every gene as a similar gene with a similar selective pressure acting on it and similar evolutionary forces acting on it, that may not be a good scenario. It may not be a good thing to do. So what we see here, which could be a very nice example of positive selection, is the history of Nicotiana rustica. You might be familiar with it if you in South America. Is that and also other Solanaceae researchers, of course, is that Nicotiana rustica is an allotetraploid species where we have Nicotiana undulata as the paternal parents, and we identify Nicotiana nightiana as the maternal parents based on whole plastid genomes. If you regard the scenario that in Nicotiana and specifically in these species, maternal inheritance of plastid genomes should be inherited. So I would argue, and I would suggest that the next step, the following phylogenetic hypothesis, should be tested with mitochondrial genomes as well. So what we see here is that the most recent common ancestor area is illustrated a dispersal event for Nicotiana paniculata in northern and southern Peru, and a vicariance of Nicotiana nightiana in coastal Peru. The overall dispersal pattern of the examine species showed a typical south to north expansion pattern from central Peru to Colombia and to Ecuador for Nicotiana rustica, to Bolivia to Nicotiana undulata. Based on this typical south to north progression scenario, what we see is that habitats located at higher altitudinal ranges were first available for colonization in the south and recently continuing northwards. And the erosion of orogenic progression, lift up, caused dispersal barriers of species colonizing these high habitats to diversify in the south to north pattern, which is very, very well known in other plant groups in the Andes. So frequent is this speciation and dispersal process is frequently followed by allopatric speciation. So for taxonomic groups currently residing through a large portion of the high Andes, a south-north speciation pattern is expected. But most importantly, this suggests that Nicotiana undulata and paniculata evolved in north-central Peru, while Nicotiana rustica developed in southern Peru and separated from Nicotiana, which adapted to southern coastal climatic regimes. And this adaptation process could be helped by positively selected plastid genes with functions such as DNA replication and photosynthesis, which might have been associated with successful adaptation, for example, in the coastal environment. The RPOA polycystron is an essential chloroplast gene, is essential for chloroplast gene expression and plant development, so it's a general triggering. It's also coming up in different plant stresses, by the ATPB gene encodes one subunit of the ATP synthase complex, which is a very, very important part of energy production, while all the NDH genes are involved in stress acclimation. So what we see here is that these genes contain positively selected sites, which in some cases are also coming with different amino acid characteristic amino acid changes for the species involved in the adaptation process. As a last slide, I would like to tell you more about structural variation and the expansion of the IR regions. So the IR regions are very important in terms of in terms of plastid genome stability in general, for survival and for the maintenance of the plastid machinery. And so far, there were no IR expanded regions described from Solanaceae, except for one very interesting, for a number of studies, actually two studies from the 80s and from later on from the 90s, which confirmed that Nicotiana acuminata might have an extended IR region. So what I did is sequence the Nicotiana acuminata species plastid genome and confirmed that indeed this IR expansion do exist in Nicotiana species, in this Nicotiana species. So what we see here is enormous, almost 12 kb of IR boundary expansion, which is going up also into the first intron of the CLP1 gene. And interestingly, this part is duplicated. Basically, if it would be a normal IR expansion, what we would see at the other end of at the other end of this genome here would be only a pseudogenic copy of the inverted CLPP gene. But instead, what we see is a very anomalous feature where we still have a truncated typical RPS19 pseudogene at the end. So how could that be? So if it would be a normal expansion of the IR region, it would be there won't be any other part at the back. So what we see here is a proposed mechanism which is mediated by, of course, the mentioned repeats, and goes through a DNA break, which is causing recombination through the appearing repeat mechanism, and that is leading to the invasion and the expansion of the IR region. So this is the story behind the Nicotiana acuminata expanded IR region, which may not be, and it is not specific to Nicotiana and to this species. So the question now comes that what happens with those genes which were transferred back, transferred from the IR to the small copy region, and from the small copy region to the IRs, because IRs go through gene conversion and concerted evolution. So genes which are found within the IR regions have a lower evolutionary rate, while genes found in the LC and in the SSC have an elevated mutation rate, and the NDS values also rise on those parts. So it would be very, and this comes to phylogenetic analysis, because what we see here is rate heterotasy. So at some point through the history of that protein coding gene, it had a different evolutionary rate, while after the specific event, it was transferred to a different compartment of the plastid genome, so the evolutionary rate could go down on that part. So it is kind of like the rate is changing through its evolutionary history, which could interfere with phylogenetic inference if your goal is making and setting these things for your research. So it's quite remarkable. I'm quite excited about it. I really love these. I really love these parts. But majorly for now, this is what I wanted to tell you. And the take-home message that you should remember is that plastid genome sequences are useful in DNA barcoding, and you can base your phylogenetic inference based on it. Mostly in smaller close related groups. And plastid genomes can be also used for investigating different hybridization scenarios and paternal relationships of different species, if we accept that they are maternally inherited. But even if they aren't, and you are looking at the protein coding genes of a mitochondrial, of a Solanaceae mitochondrial genome, you might be able to discover more important biological features for solanaceous plants. So I could just advise you to compare your nucleotide, your nuclear, your plastid, and your mitochondrial trees, because that will be the ultimate way to discover different biological scenarios. So plastid genomes in Solanaceae are, yes, they are very synthetic, also, which is also true for most Solanaceae genomes. If you look at it from that way, except for the petunia. So I expect that what we see also in the X12 clade is that you have highly synthetic regions for different Solanaceae species, close related. Then the more distant you go and you step out from the X12 clade, and then you go to, for example, Schizanthus or Salpicroa or Nolana or different small or Leucodendron, then you would see different genomic rearrangement, different chromosome numbers, and that would somehow also translate into unseen and remarkable plastid diversity. So we might be able to come to the same conclusion and to the same discovery that different that rearranged plastid genomes might exist also in Solanaceae. This comes down, according to two recent studies, why Solanaceae plastid genomes could be highly synthetic and maintain the same gene sequence order and conserved sites, is that might be that to have genome rearrangements, you need genome rearrangements in the plastids are mostly connected to Worley's and Holiday junction sites, which could, where if you have different modifications and mutations accumulating, then you have an unstable plastid genomes. So, but besides these things, Solanaceae plastid genomes may contain signs of positive selection, which would be key processes and key things to investigate different biological scenarios of adaptation. And of course, in Solanaceae and in Solanum, you have adaptive radiation, which is a very important biological process. You see that in cyclid fishes, which was recently published. So it would be very, very important to look at how these different sites and different plastid protein coding genes act during Solanaceae evolution. With these words, I show you my moving papa mug of coffee, and thank you for your attention.
Thank you very much, Peter. That was kind of yes, really, really amazing talk. So there are a lot of, we're over time, but there are a lot of questions, and we're going to do all the questions anyway. So if someone has to go, they can check later the recorded talk, if that's okay for you, Peter.
Oh, that's totally fine. I don't know how long I have been talking, but I'm very sorry about that.
No, don't worry. It's super interesting. So we're going to start with the questions. If you have a question and you need to go, just drop it in the chat. I will read it for you. Otherwise, just text like, I have a question. I will give you like to turn on your microphone and ask it. So starting with Richard Olmsted.
Oh my God.
With a comment about plastic genomes. Do you want to turn on your microphone, Richard?
Sure. That was a very nice talk, Peter. I just want to say it was really great to get caught up on everything that you're doing and others about plastid genome evolution, which is something I haven't stayed in as close touch with in recent years. So that was really wonderful. You showed the early genomes of tobacco and Marchantia and noted that the Marchantia genome was drawn as a circle and suggested that that was the first time that was done. But in fact, Jeff Palmer had been drawing circular chloroplast genomes based on restriction fragment mapping, which is how the first evidence that it was a circular genome came to be known. And the work that Arnie Bendich has done that shows that it's not always circular in nature is very cool from a functional and structural point of view, but it still doesn't negate the fact that it maps as a circle. And that is continuing true you know, even the more we learn about how it's structurally not a circle in nature, and that I think is a really fascinating aspect of chloroplast genome structure and function, and what it means, I don't know. Arnie has lots of ideas about it, but Arnie's a colleague of mine, you know, he's here at the University of Washington.
Oh my God, oh God, oh God.
40 years, and he's a real nutball. I mean, he's really bright, but he has all these ideas that, of course, you know, many of them are off the wall, but some of them, like this one, he's argued for years, and people didn't take him seriously until he did those photo videos. So that image you showed was of a whole chloroplast genomes migrating through gels, and they're radio labeled so that he could visualize them in video format. Very cool and convincing evidence that this was in fact true. So really nice talk. Oh, one other thing I'll note is that I did a lot of chloroplast genome mapping for my postdoc. That was right after the publication of the Nicotiana genome. So we took that, we designed restriction fragment. We made using restriction enzyme. We cut fragments that we could use as markers, and then we mapped very fine scale mapping of chloroplast genomes of, you know, well over 100 species. And there was one that had a large inversion in it. One species. I have to go way back into notes to find it, but I will try to dig that out and send it to you. We never published anything about it because it was easy enough to just flip the inversion for our purposes of looking for restriction fragment mutations that we were using for phylogenetic markers. And since it was just found in a single species, it didn't itself have any phylogenetic implications. But I think you might be interested in looking at it. I'll try to figure out what
absolutely, absolutely. I remember those papers from 81 and 82, and Palmer and Jeff published this in a book. It was very hard to get it, but one of my professors had it. And since these discoveries were made made at the University of Szeged and a university in Hungary, and they had connections to my university where I was working. And Paul Mulligan, I think he moved to the U.S. He's still going on, and he's still working on Nicotiana plastid genomes. And the interesting thing about, you're totally right with the nonlinear with the non-circular structure. It's just simply the circular form was discovered earlier, and Oldenburg and Bandig had this very remarkable paper in 2004 published that one ring doesn't rule them all, and it's just simply not a circle. But if you think about it, you have the flip-flop mechanism. And then you have a lot of papers, the chloroplast genome of type of papers, which are also coming out nowadays. But you see, but if you look beyond, and then you try to investigate the interesting biological features behind flip-flop mechanisms, which are happening with the SSC, with the different parts, then these things just simply could not fit into a circular genome, right? That cannot be a circle. It's a dynamic process, which is going on. It's so interesting, and yeah, definitely, Nicotiana acuminata is not the only one with the extended IR repeat. I think it's if Andres is present, then his work includes a lot of sequencing at the early branching groups, and there we have these different rearranged plastid genomes. But I would be really interested to check out what you found in back then when you postdoc. Please send that part to me. I would be very interested to look at it.
So you probably have our 1996 paper on the Nicotiana inverted repeat. Ebb and flow.
Yes.
That's where we first found the acuminata expansion.
Yeah.
We also found lots of just very small variation at the ends of the inverted repeats within Nicotiana. And that was all identified through mapping. And then we went back and sequenced across the boundaries to confirm the actual boundary repeat edges.
It's actually there. So I could that your paper is actually on my desk here. it's here. I was preparing my talk, and just a couple of weeks ago I got a sequence from our herbarium, and I just wanted to check if the junction sites for acuminata are real, and is that real? But yes, it's there. And that little part at the end I described it. It's there. It's very remarkable, but it's all there. Very, very interesting.
Great talk, Peter.
Thank you very much.
Thank you, Dick. Yes, we have another question that of Shai Faisal. He left because he had to go, but he left the question. He's asking about if there is any comparison of or a correlation between mitochondrial DNA and plastid DNA evolution.
There's not too much done with Solanaceae mitochondrial genomes. There was one very good study published with the physioclina mitochondrial DNA, and it could be very, very interesting in terms of recombination, because the size of Solanaceae mitochondrial genomes are very, very different, and they vary. And there may not be one circle for a so-called master circle to unite them all, but there are different parts which are going on, and that is certainly true for example the wide relatives of tomatoes. So if you look at tomato mitochondrial genomes, then you will see one big master circle, and then if you go towards, for example, eggplants, we are still struggling with the eggplant mitochondrial DNA with Xavier. We tried to do that several times. It just simply didn't happen. We don't know what is going on there. Oh yeah, and Stephen is writing that that Andres was very busy. Yes, of course he was. those are beautiful assemblies, and very, very fantastic things will come out of that once we are able to assemble all the different parts at the first branching group. So these different inversions and expansions will be visualized, and of course put into an evolutionary context, because that would be the whole thing and the whole idea, right?
That's great. Thank you, Peter. There is also a question of Stephen, actually, about, I don't know if Stephen, you want to turn on your microphone, ask that question directly?
Hi, thanks. Yeah, I think Peter answered that a little bit earlier. You kind of got wrapped into the other discussion about the expansion. But yeah, I was just wondering as well, because I know that there's a lot of diploid hybrids in that group. And Sandy's always finding more, I think. So yeah. Maybe there could be all sorts going on. I mean, I'm also thinking now about hundreds of different things, and in Nicotiana, maybe I'll have a chat to you about. Maybe we can find other stuff going on.
There's a very cool hybrid zone. We know exactly where it is.
Yeah.
Very, very cool. I have seeds too, kind of, but I'm not sure I have permission to grow them.
Yeah, that would be very, very interesting, because what you could really look at is that when you know the parental species, so you know the maternal and the paternal plant and the ancestors, then you could simply trace these things back, and that could also lead to the paternal leakage of different stress responses, and different stress responses could trigger paternal plastid inheritance. So if we would, for example, in Nicotiana, compare the maternal plastid genomes and the mitochondrial genomes, then it could be very, very interesting to look at, because a lot of work has been done on the rRNA repeats and different parts. So from there, it's kind of there's a very good solid background for it. What you could check using these things, and then of course there you have petunias, and I'm quite shocked, and I'm quite sad, because we only have 2 petunia sequences so far. Where are the petunia people? We have a lot of hybrids going on there.
Yes, that's super interesting. I think, yes, I don't know if Stacy had to leave or yes. Oh, here's Aureliano saying like some petunia sequences if you're interested. Aureliano, do you want to turn on your microphone?
Definitely.
Sure, yeah. Yeah, I'm in contact with the petunia community doing some bioinformatics. So yeah, if you are interested in some petunia species, I think that I can put you in contact with them. So there have been already sequenced. So the petunia genomes that we sequenced was axillaris and inflata. Yeah. But Excerpta is going to be out soon, and there are other genomes that has been sequenced or resequenced. So yeah, you send me an email, I can put you in contact with these people.
Fantastic. That would be remarkable. Thank you very much.
Yeah, indeed. Also, I mean, it's probably it's not. I'm not sure if you are interested. Also, there are a lot of GBS data too from all the different species too. So even with populations, but GBS data doesn't give you the whole chloroplast. But still, yeah, you want to jump into population studies. I mean, with this maybe have something interesting. But you send me an email, I can put you in contact with the people that have sequenced these genomes.
Okay, thank you very much. Yeah. Nice to talk to you. Hopefully we'll meet next year at the Solanaceae conference in Greece. It was postponed. I was looking forward to it, and yeah, it didn't happen because of the COVID. But it's rather doing the things through Zoom.
Yeah.
Yeah. Okay, I think we don't have any other questions, so we are finishing the seminar now. If you have any other question, of course, like you can send an email to Peter, and you can find all the information on the webpage as well. So thank you very much, Peter. There's a lot of congratulations about your talk. And next week, we are meeting for another seminar, as always, and it's going to be very exciting because it's not only a joint talk between 2 researchers, but it's also a joint talk in Spanish and English. So it's going to be, yes, so it's Joseph Jacobs and Flavio Ramos. They're going to talk about the mejoramiento del tomatillo, Physalis philadelphica, en el siglo XXI at H.M. Close, a breeding program rooted in place, focused on bringing value and linking new technology to deep agronomic practice. So yes, it's going to be very excited. So see you next Friday.